So, good morning, afternoon, wherever you are in the world today. My name's Troy, and I'm a member of the Luthi-Schulten group here at the University of Illinois. And so today I'll be talking about deciphering the influence of genome architecture in JCVI syn1. And so to kind of begin, as we're all kind of familiar with the idea of the minimal cell, there was this commentary piece that came out a few years ago that talked about this idea of minimal cell or minimal bacterial maximal knowledge. And so this idea is basically it allows us to study fundamental processes, and it makes the minimal cell an ideal candidate to do this due to the fact that it's been simplified and there are less unknown. Sorry. You heard my colleagues previously talk about JCVI syn3A, where we've developed a dynamic whole cell kinetic model. And so as Zane talked about it in his talk, there's a lot that goes into adding into the model and initial conditions that are needed. And so my work has kind of been focusing on actually the predecessor organizer, JCVI syn1, which has roughly almost twice as many genes, and trying to understand what studying syn1 can do and what kind of insights we can get. And so specifically, the process that I've been looking at is transcription, or the conversion of DNA into RNA. And so for the outline of my talk, what I will be discussing is the idea of the bacterial transcriptome, and its complexity, what I mean by that, and then how that relates to protein stoichiometry. I'll get into this idea of genome architecture, what I mean by that, and then ways to characterize it. And then I'll get into some experimental data of long-read transcriptomics that we use in conjunction with the computational method. And then at the end, I'll get into a kind of tangential experiment or work that's being done looking at the epitranscriptome. And so to kind of begin, you start with this idea that the bacterial transcriptome is complex. And so what I mean by that is there's many functional forms within the transcriptome, but also there are many processes that can undergo either to form the transcriptome or to modify it in some way. And so an example of that is differential transcription, and then some of the post-transcriptional processing that can go is folding of the messengers, let's say, or tRNA, rRNA, and then also cleavage and degradation can occur to species within the transcriptome. And what is most fascinating, or what I find most fascinating about the transcriptome is, although it's complex, it's able to produce non-trivial protein stoichiometries. So what I have here is ATP synthase to the right, very commonly found transporter within cells. It's also within the minimal cell. And what's interesting is that in the minimal cell and in other organisms, all of these genes are located in an operon. And so all of these genes are within the same region of the genome, but yet, all these genes are within the same region of the genome, and the transcriptome is complex. And so you're able to actually generate these non-trivial stoichiometries, even though that most of the processes that these genes are undergoing should be relatively the same due to the fact they're in the same genetic context. And so things like gene replication, since the genes are in the same rough region, those type of effects should be kind of similar amongst the genes within this complex. And so to kind of really understand how these things can evolve is you need to understand kind of the fact that transcription is stochastic and differential. And so what I mean by the idea of it being differential is that identical transcription events, as you can see kind of here in the schematic, can have different transcriptional outcomes. So, for example, you could have a binding event at the same promoter site or the same transcription start site, but due to different circumstances, your transcriptional outcome could be different. So you could have expression across an entire operon or expression across a specific region. And what I want to introduce also is this term that I came across within this review article, the RNA isoform, which in this context is just an RNA transcript generated from the same genomic region. And so I've been talking about the genome and these genomic regions. What's interesting within these genomic regions, if you kind of zoom in on them, thinking of the circular bacterial chromosome, is there's sequence motifs within them that have effects on different processes within the cell. So, for example, your gene open reading frame, these are the functions you're going to find within the cell. But what I find and what I think is more interesting are things like the promoter and the termination loop, which act as start and stop sites the promoter a start site and the termination loop a stop site for transcription. And so to go into a little bit more detail, the promoter itself, as I said, it signifies the start of transcription, and that is it is a site for a physical interaction between RNA polymerase and the DNA. It's been well characterized in a handful of organisms. I have some references there to some pneumonia work and also some work in Mesoplasma florum. And what's interesting is we've been able to identify it using We've been able to identify it using a sequence alignment-based approach, able to find the best potential motif for a gene, for each of the genes within the minimal cell. The same thing has been done for the intrinsic termination loops, which are sites of transcription stopping within gram-positive bacteria, and since syn3A and syn1 are derived from gram-positive, we understand this to be the most common way transcription is stopped within the cell. And so these were identified using a decision rule that was originally parameterized for Bacillus, but then has been applied to mycoides. And that work is referenced here. And also, they're in the audience. The Rodriguez group sent me these scripts, so thank you for that. Without that, I would not have been able to kind of do this portion of the analysis. And so with these sites identified, what we can start to do is looking at the transcription units. And what I mean by that is the idea of what within a given transcriptional event, the genes are products of said event. And so the way we do that is you start with the gene open reading frames, you add in the termination loop predictions, you add in the predicted promoters, and then from that you can get this idea of that if you assume that every promoter is a transcription start site and every termination site is a transcription end site, you can develop the transcription units where in this case, in this transcription unit here, the 3 genes would be transcribed, and in this transcription unit here, only genes E and F would be transcribed. And again, this is some work that was adapted, or this is work that has been adapted from some work done in Mesoplasma florum. And with all of this, we are able to then get at predictions of the transcription units within syn1 and syn3A. And what I show here is not necessarily so you can see every single transcription unit, it's just to show how complex the transcriptional unit architecture is within both of these cells, although they are minimal, and also kind of to provide some evidence that we're able to understand by studying syn1 things about syn3A. And so what I'm showing here are some regions circled where the transcription unit architecture is pretty much identical between the two species. And also you're seeing that the number of genes within the transcription units can vary from approximately 1 all the way up to the largest one is 21, which is this ribosomal protein genomic region I have identified. With these computational prediction units, the next step is to kind of try and validate it experimentally. And so to do so, we use Oxford Nanopore long-read direct RNA sequencing. And so I have here a schematic to just quickly go through it. It deals with some RNA prep stage. The RNA is then threaded through a membrane pore where an electrical current is modulated depending on the sequence of the RNA as it goes through the pore. What's important to note is this can only detect non-canonical bases in the context of RNA. The sequences are then determined for each of the transcripts, and they're mapped back to the reference genome. And then I have some example data that will be relevant for the plots I'm about to show to better explain them. And so what I have here is sequencing depth. So that's just showing you basically how many times a transcript or a nucleotide within a region has been mapped back to. So in the case of blue here, you would have, let's say, 30 different transcripts potentially that have been mapped back if all of them fall within this region. The other kind of plot that I'm showing here is looking at the isoforms within the data. And so we can actually look at where the start and end sites are for each of the different transcripts that have been read through the pore. Now going in to look at some actual data, and what we can actually do is combine the computational transcription unit predictions with the Oxford Nanopore sequencing. And what's interesting is the predictions themselves, the computational predictions actually roughly predict the sequencing depth within the DCW operon. And so if you recall, an operon is believed to be expressed in one shot. So what you would expect is that all of these genes within the DCW operon, an operon implicated in cell division, would be expressed at one time. But what you actually see is there seems to be this kind of two level of transcriptional activity. These first three genes seem to be more transcriptionally active, while these last three do not seem to be nearly as transcriptionally active, and that's determined by the sequencing depth. This can also be determined in the computational prediction units, where you have multiple transcription units that contain these first three genes. And so in some toy model simulations, if you run simulating based on the transcription units, you actually get out this like rough difference you see in the sequencing depth with respect to the abundances between the genes within the operon. And why at all is this important? I started off talking about ATPase. Well, the proteins within this have to be in a certain stoichiometry to do their function. It's been talked about already somewhat during today's talks, is FTSE and FTSA. And so they're implicated in cell division, as I mentioned, and they need to be in a 10-to-1 ratio. So that's these two genes here. And so what you can see is this is maybe a potential way where gene 522 FTSZ is being expressed more, and so that's more likely to be translated, which could mean more likely for the protein ratio of 10-to-1 to be achieved. Moving forward so I just showed kind of an example where the computational prediction units and the Oxford Nanopore data agree really well. Here's an example where they don't as much in the sense of this beginning region here. So if you look across the ATP synthase operon or genomic region, you see that most of the expression or the sequencing depth is consistent throughout it, indicating that the expression is consistent throughout it, except for at this beginning region here, which is not predicted by the computational transcription units. The other kind of thing that is important to note here is that this protein here needs to be in a 12-to-1 ratio with proteins downstream of it, or upstream of it, I'm sorry, or no, this would be downstream based on this strand. I apologize there. And so as a result, if you were to do those same kind of toy model simulations, there seems to be no way to get at the protein stoichiometries or get at something that could maybe lead to the protein stoichiometries. And so this is where we start to look at the isoforms or look at maybe more post-transcriptional activity with these genes that ends up leading to the stoichiometry you see in the complex. And so if I show here the mRNA isoform start and stop sites across this region, what you notice is that although some of them do seem to go across the entire genome and actually start in and outside of the operon, a good portion of them are at this beginning region, which are only these first two genes here. Another thing to kind of notice, and I have here, is that a lot of the transcripts, if you look at maybe, let's say, this black line here, is it stops in the middle of 792. That could be from the experiment itself, but also this could be the idea that transcripts are sometimes incomplete, meaning all of the genes that are found within them are not fully encoded. And this is something that's been observed recently in E. coli. The next thing to look at is the mRNA isoforms in terms of the abundance of the genes. So what you can do is kind of assign each of the transcripts to the different genes and look at their relative abundance. And what's interesting here is, as you'll notice, there's a lot of different variants of the different isoforms. And so you can determine the genes that are within the isoform by just kind of going down. So in this one here, which has a relatively high abundance within this region, it's containing these two genes here. What's interesting is I talked about that beginning region having activity that we were unable to detect using the computational predictions. What some new data has shown from some work by David that's been mentioned, these structure predictions, is that this gene here is seemingly ATPI, which is believed to be a regulatory protein for this operon, or has some regulatory proteins within, or has some regulatory capability, I'm sorry, within this operon. Kind of switching gears a little bit, another thing that you can kind of gather from comparing the computational predictions with the Oxford Nanopore is looking at unintended consequences of the genome reduction. So John kind of mentioned this at the very beginning when he spoke. So there's a protein HUP-A that is a histone-like protein that's been implicated in persistent supercoiling. And so it was identified that syn1 doesn't have persistent supercoiling based on work that Ben had kind of talked about or touched on. And what's even more interesting is that when you look at a comparison between syn1 and syn3a, is that the relative expression has drastically decreased going from syn1 to syn3a. So in syn1, there's about 3,000 copies, but kind of more importantly is that it's one of the most expressed proteins. It's expressed more than roughly 99%. I think it's like the fifth most abundant protein, while in syn3a, there's 30 copies, but again, more importantly, is that it is only expressed more than roughly a quarter of the protein. So its relative expression has changed. And what has changed between syn1 and syn3a? Obviously, genes have been removed, but more interestingly is that within going from syn1 to syn3a is this gene here, 349, has been cut out. And so if you actually look at my slide's not progressing. If you actually look at the mRNA isoforms, their start and stop sites within this region for those assigned to gene 350, is you actually find that 90% of those genes start or fall within 349, and this doesn't even include these few that you're seeing here that are potential read-throughs that would go from 348 to 349. And so what this is kind of showing us or telling us is that you can have unintended consequences by removing a gene that impacts the genome architecture of the cell, thus kind of changing expression for other genes that you maybe did not intend to modulate in any way. And kind of to wrap up, some kind of ongoing future work is we're looking to try and go beyond the transcriptome, and so to this idea of the epitranscriptome, which is just referring to the modifications within RNA in the transcriptome. So I said there were many functional forms. Those functional forms then can have various states, would be the simple way to kind of describe that. And so there are many different types of modifications. A common form is methylation. And so what we've been looking at is specifically the M6A modification that you find in tRNA. And so what I'm going to do now is explain kind of how we do that, which is using EpiNano. And so this was published here by the Novae, I think is how you say it, group. And what it is it's a machine learning algorithm that takes in the base calling errors from Guppy. So recall that I said it can only do canonical bases. If a base has been modified, it's not going to detect that. But what you can do is look at the per base quality, the mismatch frequency, and the deletion frequency of the reads as they go through the nanopore, and construct a model that is able to classify if a base has been modified or not. And so what I'm showing here is an example from the paper of the summary of it. And so when you have unmodified, or as it goes through the pore, unmodified bases are going to be read as their canonical bases. But in this case here, we have a modified base. It could be, instead of being read as an A, it could be read as a C or something like that. And so they basically take into, or they use this ability slash strategy of the base calling is referred to determine if a modification has occurred. And so what I'm doing is using previously trained models and applying that algorithm to detect modifications within syn1. The big thing to note, and you'll see this on the data is I'm about to show, is it's very prone to false positives when not used with a wild type and knockout sample. And so if I go forward, the M6A detection results, there's a typo, sorry, for leucine. And so what you're seeing here is I've done it with two different kind of approaches. What's important to note is that there are many false positives. So anytime you see kind of these numbers or these green lines, it's indicating that that base has been modified. The other thing is we're expecting the modification to be somewhere in this region. Based on it should be at position 37. Or roughly position 37 within a tRNA. And as you can see, there are modifications outside of it. The positive thing is that in some improvements I made using the more modern base callers, you do see less modifications being detected, so less false positives, but this is something we need to verify. And so actually, if there's anyone in the audience who has any sort of understanding of methyltransferases or the ability to work with them, we would very much like to kind of talk to you about potentially trying to generate a wild type and a knockout sample to kind of do this analysis in a more appropriate manner. And kind of with that, I just want to conclude. So to just kind of re-summarize some of the points. The bacterial transcriptome is very complex and has non-trivial protein stoichiometries that can evolve from it. I don't know why my slides aren't going forward. Computational predictions of transcription units based on the genome architecture predict the Oxford nanopore results. The Oxford nanopore direct sequencing allows for the detection and characterization of the bacterial transcriptome, and this is in multiple ways. So the mRNA isoforms present, the epitranscriptome itself, transcriptional activity within a region, and then some work I didn't show is actually we're able to identify new regulatory elements such as riboswitches from comparing the computational predictions with the Oxford nanopore, and where we see termination or something like that occurring where we don't expect it, we then can go in and kind of try to better understand why this is occurring, especially if it's happening consistently. And then the last kind of point is that genome minimization can have unintended consequences on relative gene expression. And with that, I would like to acknowledge my group, past member David, and then current members Zane and Ben, all of the collaborators, John Glass and the JCVI, Pratap, who did and helped me with some of this work, and then Chris Fields here at UIUC, who was very helpful in discussions about kind of this analysis. And with that, I'll take any questions.
Really nice, Ben. So you showed instances, particularly with the ATPase, that suggest that
my camera wasn't
based on transcript levels, the protein stoichiometry that we observe isn't borne out by what you see with the transcripts. Thinking about that, you have the mRNA entering the ribosome, and then one protein gets made after another, right?
Yeah. So I guess go ahead. I think you have something more to say. Sorry.
So what I'm wondering, in viruses, we often see instances of internal ribosome entry. Could that be going on? So if the isoform levels don't explain the protein stoichiometry, could we be seeing instances where irises are allowing this to take place?
Where viruses are allowing it to take place, you're saying?
No, irises, internal ribosome entry sites.
Irises being as I understand it from what you said, is like translation being able to occur like mid-transcript is what you're saying?
Yes.
Kind of?
Mm-hmm.
So I would think so, based on the fact that my understanding is that polysomes can form dynamically, and they can form on messengers as they're being read. And so if a ribosome could form over top of a messenger, then that could be one way. The another thing that I didn't mention is that there are some cleavage events that don't necessarily have to do with degradation, but just have to do with the maturation of the transcript. That's been understood in Bacillus for some of the proteins, or one of the genes within, I forget the exact subunit, but one of the genes within the ATP synthase operon. And so I think it's more likely that it's things like that, where transcription or post-transcriptional activity or processes are what are leading to it, more than just the transcription events themselves.
Okay.
So it's the idea, the way I kind of think of it is, it's easier to make a bunch of things and then kind of figure it out than make sure that every single time you make the right thing in the right ratio. Kind of akin to like cooking for people. It's much easier to make more than try to make sure you guarantee that you can feed everyone the exact amount.
Okay. Other questions?
Hey, Troy. That was a really interesting talk. I had a quick question about that kind of last point you had at the end. It's mostly out of curiosity than anything.
And this is John Sanford from UAB.
Yes, sorry, I should have introduced myself. Forgive my manners. Did they, or when you look for riboswitches and whatnot, have you noticed anything particularly, or is there anything like noteworthy about it? Like, is there like a high degree of riboswitches relative to the genome, or is it more just kind of typical?
So we've only been able, I think it's 3 or 4 that were previously identified. We were able to identify 2 more from using like a search algorithm, and then we kind of validated them comparing. What I'm kind of currently doing is looking at it more in a more kind of systematic way, using the data, looking at where we see transcription stopping or being altered where we don't expect it to be, and then going kind of back and then seeing if there could be a potential riboswitch in that region. We haven't been able to find any more, but I really think there are more. Based on there are more in other organisms that we haven't been able to specifically identify for ours, but it seems to be a very efficient strategy the cell comes up with to modulate and be able to sense its internal environment, because a lot of these riboswitches bind something and then that will turn on their activity or stop their activity. And so because of that, I think there are definitely more there than what we've been able to find, which is on the order of like 5 to 8, let's say. Thank you for the question.
Thank you.
Anyone else? Ben, your analysis of the nanopore data really makes me happier about wanting to use the direct RNA sequencing.
Yeah, it seems to have its nice perks. There are downsides, obviously, to everything, but I think the Oxford Nanopore has a lot of upside at this point with what we've been trying to study and uncover.